Good morning, everyone. First, I'd like to start by congratulating Chris and his team for really putting together a tremendous forum here. It's really been a lot of fun, and I think today will continue to be great. This morning, we're going to talk about Alzheimer's disease, and the theme will be Alzheimer's Phoenix, the idea that Alzheimer's has had some tough times in terms of trials and setbacks, but this past year or so has been particularly exciting with new enthusiasm and hope for the basic treatment and prevention of the disease. So we have a great panel. I can't think of anyone I'd rather have sitting here than these folks right here who've been working in this field for way too long. Yeah. So I will just introduce Dennis Selkoe, who I'm sure you all know, who's who's a professor of neurology at Harvard and also co-director of the Center for Neurologic Diseases at Brigham and Women's. We have Dale Schenk, an old friend, who's now CEO of Prothena. I think we're all old friends here. And Mike Hutton, who's now CSO of Neurodegenerative Diseases at Lilly. Roger Nitsch, who's sporting a very nice tan, having just come from Italy, and who's director of psychiatry research at the University of Zurich and also with Neuromune. And finally, my old friend Brad Hyman, who's director of the Massachusetts ADRC and also professor of neurology at Harvard Medical School. So let's begin with Dennis. Dennis, when I met you, I was a student, and you were barely a junior faculty member. Right. And at that time, we barely knew about what amyloid was made of. I remember the first time I saw you speak at MassGeneral, we didn't know what amyloid was made of yet. And you had the stone with the sword in it because you were spending a lot of time trying to get amyloid dissolved to figure out what it's made of. And so you've been in this longer than anybody in terms of the roller coaster ride of amyloid, and have been a great champion of the amyloid hypothesis. And these are great days for you, given what's happened recently. So maybe you could just start by telling us about that journey and the roller coaster ride and where you think we're going.
Great, Rudy. Well, thank you very much. Delighted to be with you this morning. So it has indeed been a roller coaster ride, but I have a real sense that we're up on the top, seeing the valleys and mountains ahead of us quite clearly now. And in so many ways, you wonder why it took our field so long to get to a target that was really therapeutically valid. It's surprising to me that George Glenner's concept in 1984, and really before that, he already spoke about the concept that an amyloidosis of the brain might be the driving force behind Alzheimer's, and he quoted the information about Down syndrome, which was a very powerful clue. And yet, our field, the field of neurobiology, applied neurobiology, had a hard time accepting the notion that a small hydrophobic peptide would be the true starting point for the disease. And even today, we don't know that. You're going to hear from my colleagues how that concept has to be validated. I think that in the periphery, in systemic organs, physicians knew for a long time, for 100 years, that amyloid buildup was a bad thing and could lead to disease. But in the nervous system, for some reason, I remember friends whose opinions I very much respect would say that Alzheimer's will turn out to be a remarkably complex signaling abnormality of synapses that will feed back on it, and it'll be very hard for us to see the beginning of it. My opinion is, and it's still an opinion, that what triggers that synaptic alteration is the buildup of small soluble oligomers of A-beta. And that really is the theory that Glenner put forward, and that a few of us in the field, and I think all of us on this stage ultimately bought into that, thought was going to be the starting point. And today I think you'll hear from the panel that we're there in many ways. We have clarity about this. One of the things I like about the amyloid hypothesis and the idea that A-beta dyshomeostasis could drive this complicated neurodegenerative disease is that this theory, as you would know very well, Rudy, from your pioneering work on Huntington's, really came before the genetics started to validate it. So we knew from the biochemistry that was done from the molecular pathology of Alzheimer's that A-beta was a peptide that built up in vessels and plaques. And there came the amyloid hypothesis. And then in 1990, 1991, 92, we got genetic validation. So in short, it feels like a rational biomedical thesis. It hasn't been fully proven, but it looks like it's coming very close, as you're going to hear from others today. And I think the roller coaster ride basically is what we expect in complex chronic diseases, that it takes 3 decades to figure out if you're on the right path with a lot of Sturm und Drang along the way.
Great. So, Dale, you're the, I guess you would call you the godfather of immunotherapy of Alzheimer's disease.
I am Italian.
Yes.
A little bit,
Are you half Italian?
A little bit. Yeah, well,
yeah, a little bit. You look Italian.
Yeah.
So maybe you can tell us a little bit about the history of immunotherapy in this disease, the ups and downs, and how you're feeling. Are you optimistic about the potential for going forward, and what are your own thoughts in terms of immunotherapy also for other neurodegenerative diseases now that you're working with Prothena?
Yeah, no, absolutely, Rudy, and thank you very much. This is a great forum to talk about this. Well, so yes, let's talk a little bit about the A-beta hypothesis and immunotherapy. Way back when Dennis had first suggested, and Glenner and a few others had said, A-beta may be the root cause of the disease, we were tasked with, well, okay, that's very interesting. How do we make a difference for patients? And I won't go through all the different ideas that all of us have worked on over the years, but one simple idea that we had way back when in the late 90s was let's just vaccinate with A-beta itself to create an immunological response. And hopefully that might have an effect on the brain. And there's lots of reasons it shouldn't work. In fact, many, many reasons it shouldn't. But to make a long story short, at least in the animal models, it did. And I'll admit, to our surprise and everybody else's surprise. And then we took it one step further and said, okay, what aspect of the immune system was responsible? And it turned out it was the antibody response that we generate. So basically it means that antibodies to A-beta, back then, going back to now, let's say 2000 is when we published that, we said, okay, let's see if we can help patients. And we've collectively spent 15 years doing clinical trials to sort that out. And I'll just summarize a little bit of that, but I'll talk mostly about the early piece when we first started to try and help patients with this disease. In the year 2000, although the disease could be diagnosed, it was a clinical diagnosis. So it was very hard to differentiate prodromal from early from moderate. So we couldn't really diagnose very well. In fact, we were told, ironically, by the FDA, we weren't allowed to just treat early patients. We had to treat everybody. That's an interesting point that'll come up later as we talk about where we're at now. We also had no validated imaging marker, so we couldn't look in the brain very well. We had a little bit of that going on, not much. And then finally, with our first clinical trials, we hit vasogenic edema, which I won't go into the details of, but it meant that we could only dose at a very, very, very low level. So we did our antibody clinical trials with those caveats, and it was a very long, complicated, interesting, but ultimately unsuccessful story, at least with the antibodies and vaccines we were working on. But fortunately, the whole biotech and pharmaceutical industry by this time had also launched multiple programs, and more importantly, I think, the clinical space had advanced to the point that you could diagnose, you could image, and you could understand the safety issues. And so ultimately, that brings us full circle to now, which is with the recent data from Biogen, and we're going to hear more about that antibody, where they've got all those things right, I think. We're seeing the first positive results with immunotherapy, both in terms of reducing pathology and in terms of clinical response. So I'm very bullish. I think Phoenix is a fantastic name for this forum. And I hope the message gets out to patients, Alzheimer's patients, that there now is very real hope.
Great, thanks, Dale. So Mike, you began in this field as a tau guy and became world famous for finding the first mutations in tau, leading to frontotemporal lobar dementia. And now you're forced to have to work with an amyloid antibody, one of the best ones, solanezumab. And I'd like to ask you, we'll talk about tau and the other pathologies a little later, but I'd like to get your perspective on SOLA and the A4 trial that's going on around the country and here in Boston. And what do you think the prospects are?
Thanks, Rudy.
Or anything else you want to talk about as well.
Okay. First, let me start by saying that, as you've heard from Dale and from Dennis, I think it's an exciting time to be working on Alzheimer's disease, and I do think that we're actually now very close to disease-modifying therapies. These are molecules that will actually slow the progression of disease. They won't just simply treat the symptoms. And we think they're working by essentially targeting this key part of the underlying mechanism, namely the amyloid plaque pathology that so many of this panel have contributed to down the years. However, why are there now such optimism, given the fact that we've had failures of similar potential mechanisms in recent years? So why are we so optimistic? And I think, first and foremost, I think we're starting to understand the reasons for some of those early failures, be it poor target engagement or because we selected the wrong patients for the clinical trials. But in addition, I think the other key point is the optimism is based on the molecules that we actually see in company pipelines that are expected to complete phase 3 trials and indeed to launch within 5 years. These, generally speaking, improved molecules that more effectively target A-beta and the beta amyloid pathology. So as Rudy's mentioned, I'll talk about solanezumab and then a couple of other things in a minute. So firstly, just talk about SOLA. It's Lilly's own antibody. It targets soluble monomeric forms of A-beta. As you've probably heard, the molecule failed to reach its primary endpoints in its initial phase 3 trials. However, what we did see was in a subpopulation of mild AD patients was a marked approximately 34% slowing in the rate of cognitive decline. And this really provided the first clinical evidence that targeting A-beta could slow disease progression, but critically, if we can treat patients early enough. SOLA also displays an excellent safety profile, which I think will be important. And some of the patients have now been on the drug for over 5 years as part of the continuing open label extension. And we're now testing solanezumab in a more targeted phase 3 program. It's only in mild AD patients where we've confirmed amyloid pathology either through an amyloid PET scan or through a CSF biomarker. And we anticipate that study, that phase 3 study, will read out in the second half of 2016. As Rudy's mentioned, solanezumab is also being tested in asymptomatic at-risk patients as part of the A4 study. In the A4 study, we're targeting patients that have amyloid plaque pathology but have yet to develop clinical signs. And then the second at-risk trial is in patients who have mutations that we know will cause Alzheimer's disease, and that's as part of the DIAN trial. Now, those trials will take a lot longer to read through, probably 2019 or later, but nonetheless, there we're moving even earlier in the disease course with the expectation that these molecules will have greater effect if we can get to that early stage. I think the other molecules, just to finish off, really, if I've got time,
Please,
that I'm excited about are the BACE inhibitors. These work by inhibiting the production of A-beta and really can produce profound reductions in the levels of brain A-beta of up to 90%. And as a result, these are probably now the most competitive. This is probably now the most competitive target in the industry, with multiple companies now in clinical development, including Merck, which has the leading molecule, Lilly and Janssen, among others. And we do now expect to see phase 3 data readouts that it's expected from 2017. So again, a very exciting target, a very exciting set of molecules that produce a really profound pharmacodynamic effect on A-beta, and we hope, therefore, the downstream effects of that pathology. Behind the BACE inhibitors, there's, as you'll hear about from Roger in a second, there are a bunch of other different molecules, of which aducanumab, the plaque-specific antibody, is one example. I won't say more about this here. And as Rudy has mentioned, my background is very much in tau. So I think the other great thing that we've just started to see, just literally in the last couple of years, is the appearance of a number of molecules that target tau and the neurofibrillary tangles, the second pathology of Alzheimer's. In particular, tau antibodies that are designed to block the spreading of tau pathology are starting to move into clinical development. And one reason why that's particularly exciting, of course, is that it may allow us to, at some point in the future, combine amyloid and tau therapies to produce an enhanced clinical effect. So I'm excited.
Great. Thanks, Mike. So, Roger, we've all known you a long time, and you've always done things a little differently, in a good way. And you've been working on a very exciting immunotherapy. I don't know if I can say this word, aducanumab. Aducanumab. Sounds like a Genesis song, which was a good song. But Roger, you took a different approach, as you know, and maybe you could talk about the history of this exciting antibody and what you think the prospects are for hitting amyloid early.
Well, thanks, Rudy. These are exciting times indeed. A different approach indeed, and this is an antibody that has been shown recently by Biogen to reduce amyloid in a dose-dependent manner and also a time-dependent manner. So, in the highest dose, when you treat the patients for 1 year with 14 doses of the antibody over the course of 1 year, you get almost 100% reduction in the amyloid. So it's absolutely exciting to see how an antibody can remove this material that takes maybe 15, 20 years to build up within 1 year. Absolutely exciting. Also exciting, of course, for a guy who has looked at these things in the laboratory before, that the effect was much greater than what we were able to see in mice. So it seemed like it's easier to treat patients than transgenic mouse models, which is also a very interesting lesson that we learned here. And of course, what is also extremely exciting is that for the first time, this reduction in amyloid correlated with a clinical, also dose-dependent, time-dependent clinical stabilization of the effects. It's a small trial, phase 1 trial, so we have to be cautious about the interpretations of the results. And I think Biogen is taking a very bold move now to move directly into 2 substantially large phase 3 trials, and we'll have to wait and see how that turns out. Now, coming back to your question, we took a different approach to making the antibodies. We call this approach reverse translational medicine approach. This is based upon the hypothesis that old people recognize, or in fact their immune systems recognize the deposition of protein aggregates, not only beta amyloid, but also tau, synuclein, TDP-43, C9orf, and deposit in their bone marrow B cells that have a memory for these targets. And the underlying idea for this hypothesis was that the structures or the conformations of these protein aggregates are different than their physiological precursors, so there's probably not much tolerance in the immune system. When we looked at that, the results were that about 80% of healthy aged individuals have B cell memory against beta amyloid, more than you'd expect. Now, of course, with this finding, you don't have an antibody automatically, because this kind of B cell memory doesn't necessarily make molecules that have drug-like properties. So we had to initiate a huge process of screening many millions of B cells in order to find out the very few ones that encode antibody sequences that in fact target our structures, in this case amyloid, pass through the blood-brain barrier, don't interact with any other tissues in the brain. So this is a huge process until you end up with a molecule like this one, aducanumab. We now use this approach also for other targets, tau, synuclein, TDP-43, even IAPP in diabetes. So this technology platform is universally usable for many different things, and we're so pleased that the forerunner aducanumab is now showing such fantastic initial clinical data.
Great. Thanks. Thank you, Roger. So Brad, you know, you're last because you're such a wise guy, you know, and a wise guy. And so we've been focusing so far on A-beta immunotherapy, which is, I think, driving a lot of why we're calling this session the phoenix of Alzheimer's disease. But for the most part, we believe we have to treat early, maybe even prodromally, given how early amyloid is accumulating in this disease. How would you picture, let's say some of these immunotherapies are successful, and how would you picture treating your patients or treating folks to prevent this disease? What would you, just looking ahead, what would the scenario be for trying to prevent Alzheimer's disease? How early would you have to start? What would the challenges be?
Thanks, Rudy.
Yeah.
I'd like to pick up on some of the points that Dale especially made, that when the trials were started, we started treating patients who already had severe symptoms, that they came to their physician, they came to their neurologist because they no longer could carry out activities of daily life. They couldn't remember what their spouse's name was. They couldn't do things that were really obvious to anyone, that there was a major brain problem. And when you look at those individuals by MRI scan or at autopsy at their brains, they've lost something like a third of the substance of the brain. And at that point, that's really organ failure. So that's the equivalent of kidney failure, needing to be on dialysis, or heart failure and needing medications for that type of problem. And the trials at that point saying, well, can we bring that back? Can we restore that function? That was a pretty high bar to try to overcome. Now, over the course of the last decade, as Dale mentioned, with new CSF, spinal fluid markers, new PET scans, better clinical detection, we're able to not say, oh, well, you know, you've reached a point of maybe, I hope not, but no return. But instead, back up the disease process to the point where very early on, there are the changes in the brain that over the course of the next however many years will lead to the cognitive symptoms, but before the brain itself has been so damaged that it can't carry out its normal functions. So we'll be able to have PET scans, we'll be able to have spinal fluid tests, potentially even plasma tests. We'll be able to have more sensitive cognitive instruments, so that if you go to Stop and Shop and you forget something that Dora told you to bring home, not only will you be in trouble from Dora, but you'll have to come visit me in the clinic. It'll be great to see you there, Rudy.
Thanks, Brad. Appreciate that.
Yeah. So you know, it's getting to that point early on where I think there's a consensus in the field now that amyloid is important to initiate the disease process. As Mike mentioned, it seems as if tau plays a very important part in the progression of the disease, in having it get worse from initial symptoms, even initial sort of vague memory symptoms, to broadening out across different cortical areas to affect language and judgment and personality and so forth. And it targeting those different aspects of the disease, targeting the initiation of the disease with one class of drugs, targeting the progression of disease with another class of drugs, and I hope eventually even targeting that organ failure with a different class of drugs, of drugs that might be able to protect remaining synapses and restore their function to some extent, is the future. So much like we do in heart disease, much as we do in cancer, that we will have different classes of drugs that target different aspects of the disease, and potentially even heterogeneous types of disease. It's not clear to any of us that it's all exactly one disease, and it might be that there are different genetic underpinnings that will lead to one type of therapy versus another. We're already seeing that emerge in the Biogen trial, where substantially different doses were given to people with one type of ApoE genotype compared to another type of ApoE genotype. And I think more of that, and you know, your own work, Rudy, points to that more of that sort of heterogeneity of the population is going to impact how we have treatments for the disease. So I think 5 years from now, 10 years from now, whatever, we're going to be treating patients based on their specific symptoms. We're going to be treating patients based on their specific biology, and we're going to be changing that and evolving that treatment as the disease progresses.
Great. So continuing on this theme of, cholesterol and heart disease and a statin for Alzheimer's by hitting amyloid, I'd like to get next to the topic of, do we picture that we're going to have to treat amyloid in someone, let's say at 40 years old, you have a scan and your amyloid and your amyloid level in your brain is so high that there's some alarm bells that go off, and you say, well, we need to stop bringing your amyloid down now, even though you're not showing any symptoms. And what would that scenario look like? Will an antibody be feasible given IV injection, given the cost of an antibody? What are the other alternatives? We heard about BACE inhibitors. We also know about gamma secretase modulators. So how would you picture, Dennis, putting a prodromal person who's just had a scan, who's perfectly fine, and their amyloid's high, what are the options going to be 10 years from now? Antibody, BACE inhibitor, gamma secretase modulator, and what will the challenges be in terms of safety and how long that person would have to be on such a therapy?
So I think as a neurologist, I expect that we are going to practice Alzheimerology that way. We're going to get a risk assessment profile, the way that we know people do in cardiac disease and even in cancer, where genetic factors can be identified at childhood that give you a very high risk, and you start planning ahead. So for a chronic disease like this that's so common, 40 million worldwide, and the number is probably 3 times as many as that because there's this long prodromal period of lesion buildup, we're going to do genetic testing. I think there's going to be an Alzheimer panel or so. You're a world authority on this, and I think we'll be very inclusive, many different genes, and we'll learn about what their different risks are. We're going to have biomarkers, and I think Brad made the point about plasma. One of the things that this audience and all of us have to take away from a session like this is that we need to do better in figuring out biomarkers that are readily available. Now, I happen to think that spinal fluid analysis is not done sufficiently in the United States, and we know many of our colleagues feel that way also. In some European countries, it's much more common, everybody who comes in. So I think in the future, plasma tests will be done, and we need to really work much more diligently on that. Imaging procedures that are always expensive but are going to get less expensive, gene risk profiles, and we'll create virtually a numerical score that will tell me when I was 40 or 50 what the risk is. And then we'll choose, hopefully from several small molecule and large molecule therapeutics. I think it's not surprising that antibodies are in the lead right now. Dale's pioneering work and the work that Mike and Roger and many others have taken forward to get antibodies that are completely specific. The adverse events aren't really there. There are adverse events called ARIA-E, but they're a sign that the antibody's working. So I think the antibodies are going to be very good for clearing what's already there, maybe at 40 or 50, but small molecule therapeutics and even lifestyle changes, increased exercise, increased doing what this audience is doing, trying to listen to ideas that may be different than what they deal with every day, that's going to make a big difference. That's where I think the clinical treatment is going to be.
Great. So Dale, you've invested and continue to invest your career in immunotherapies, and part of the one concern will be the cost of immunotherapy, having to use it early on for prevention, whether this is immunotherapy for Alzheimer's disease or Parkinson's. What are your thoughts about buy-in from the government and the cost of covering an antibody that could be quite expensive, where you might have 5 to 10 million people in the country who might want it for Alzheimer's, plus the other immunotherapies you're developing for other diseases?
Right. Well, so there's a couple comments. First, everything always goes around to risk, reward, et cetera. And so it depends what else is available. Let's face it. And I think we have to separate too, just to augment what Dennis said and then come back to your question too. There's prevention and there's treatment. And there's always different landscapes. And you mentioned immunotherapy. In general, we call it protein immunotherapy now. We know with cancer immunotherapy, you're trying to stimulate cells to go after the cancer. This is protein immunotherapy. We're trying to stimulate antibodies to clean up the the amyloid. And you're right, there's a lot of different amyloids besides A beta. There's tau, IAPP, Roger, you mentioned several of them, synuclein. There's light chain amyloidosis, which is what we're working on. If it's the best there is, and this is true of all medicine, if you have a treatment that makes a difference to you as a patient that is better than other alternatives, it makes sense to use it, quite clearly. I think in terms of antibodies per se, they will be used primarily for treatment because of the points you make. The exception might be, and we'll see, maybe in true prevention there might be a role for them. I don't know. BACE inhibitors obviously in true prevention offer a huge opportunity. But it's always about risk-reward. If there's nothing else available, if you're a patient, you're going to want it and you're going to want to use it. So, and I think it's a dynamic that we continue to discuss between biotech, pharma, and the government, et cetera, that we need to continue to discuss. It's a big question you've asked.
Yeah. I'm going to jump to Roger for a minute because we had a discussion at a meeting at the ADPD meeting in Nice about the trade-off of the cost of an antibody, how long you'd have to use it, and nursing home costs, the cost of care. And you had some very interesting insights when we spoke about this. How do you see this? How long would someone be, let's say the antibody becomes cystatin for someone who has too high an amyloid level. Are they on it for 30 years, 40 years? Are they on it just for a while? What is the cost when you have to treat 5, 10 million people this way versus the cost of nursing home care? How would the government look at this in your mind?
Right, so I think you have to look at the global picture. The largest cost right now is the cost for care. You know, when you think about an Alzheimer patient in the advanced stage and to take care of him, say, at home, you need 4 people in 3 shifts. That's the price of your care. So if you're able to basically move the time at which someone has to have that care for about 18 to 24 months, I think pharmacoeconomics shows that this will be already cost effective. The problem is that the payers are not always the same. So there needs to be a coordination among payers who take care of the care cost and the treatment cost. And coming back to your question, I think with the advent of specific molecular imaging tools, like the amyloid imaging, the PET imaging, the tau imaging coming up, the synuclein imaging coming up. The neurologists in the future will have, in 5 years, will have the possibility to test for the presence of the pathologic protein in the brain, and that will trigger the treatment decision. So if you have amyloid PET positive, you will get an amyloid treatment. If you are tau positive, you will get a tau treatment. If you're synuclein positive, you will get a synuclein treatment regardless of the clinical symptoms of that disease. If you have a combination, you're probably up for combination treatment. So it could be as simple as that, in fact. And your question is, how long should that be, used? Do we have to use the antibody for 30 years? I don't think so. If 1 year treatment is enough to reduce amyloid to a normal, pathological level, then we may not have to use the high doses that are required for removal in order to prevent further buildup. So it's possible, and studies will have to show that, but there's several possibilities of study outcome. One is you're just resetting the clock by 20 years, and it will take 20 years again for buildup. The other thing is it could come back faster, but you may be able to reduce the dose to a low maintenance dose, which of course will cut down cost also significantly. So right now, my estimation is that we're going to have treatment cost at least for the initial 12 to 18 months of treatment. And then studies will have to show how that will continue. Yeah. And I think in terms of the government or whoever is running the payer systems, these guys need to sit together now and think about the coordination and the balancing of the various effects on the healthcare systems.
Right. In the trial, in some cases, you knocked amyloid back down to baseline.
Right.
So you can imagine if you hit early enough, you had patients where the amyloid levels were not exceedingly high, knock them down to baseline, look at them again a year out, 2 years out. So this is very different than a scenario where you're taking a Lipitor every day to keep cholesterol down. In this case, it's management as needed, which could mitigate some of the cost. And perhaps the same thing with SOLA.
We were just saying we could maintain with a BACE inhibitor potentially as well as the other.
Yeah. So let's talk about BACE inhibitors. Now, BACE, and Dennis knows this, BACE has other substrates. BACE, beta secretase hits neuroregulin, which makes myelin. We know that beta secretase hits the sodium channels and controls both the beta-2 subunit, alpha subunit production. And there are other substrates, as Dennis's lab and other labs have shown. Are we going to have, now we saw that with gamma secretase inhibitors, you hit gamma secretase with a sledgehammer and there are problems. And gamma secretase modulators, like the type that Steve Wagner and I are working on, Dennis is working on with Mike Wolf, are more APP specific, and we're hoping that those will be another way to have a little white pill that lowers A-beta. What are your thoughts about beta secretase inhibitors? I'll ask a very pointed question. Are we making the same mistake again by hitting beta secretase with a sledgehammer, like the mistake we made with gamma secretase? Do we need an APP-specific beta secretase inhibitor? Do you have any concerns about safety going forward on BACE inhibitors?
I think the simple answer is at this stage we do not know the answer to that question, Rudy. I mean, obviously a couple of BACE inhibitors have failed because of safety issues, but we think been due to off-target problems rather than on-target safety. And I don't think we have seen anything from the clinical experience thus far that would suggest that there are on-target safety problems that would prevent us from bringing a BACE inhibitor forward into the clinic and ultimately to treat for Alzheimer's disease. I think crucially, I don't think you necessarily need to inhibit BACE completely. You may be able to maintain some function which is enough to mitigate against the safety risks. But as I say, I mean, it'll be an empirical answer. I mean, so far, there really isn't the clinical evidence to suggest it'll be a major problem. The Merck trial's been running now for well over 2 years. I think if there had been a huge safety concern there, we would have probably heard about that by now. So I am optimistic that we will see BACE inhibitors become part of the way in which we treat Alzheimer's patients. The question is the extent of BACE inhibition that ultimately will be tolerated, and the extent to which we use different doses, as we've just been discussing, based on whether you're talking about prevention versus trying to treat patients who already have some kind of symptoms.
Dennis, do you agree, or do you have more concerns, less concerns about BACE inhibitors?
There are 100 substrates known for BACE, not all of them completely validated, but looks very likely. It makes perfect sense. These are highly conserved evolutionary mechanisms. The same is true of gamma secretase, presenilin, necessary for life in all metazoans. So it's likely that we'll see adverse events for one or another compound. The question is, are they target related, as Mike said? If they're off target, if they're idiosyncratic, well, then that compound may be put aside. Another one, if they are on target, if they're because we're inhibiting other substrates, which was the case with semagazostat, for example, which was problematic because its therapeutic index versus Notch was not large enough, then that's going to be a problem. My guess is I would favor what Mike has said, that chances are reasonable that we can adjust doses of BACE inhibitors, that if there are on-target side effects that are adverse, we'll find a dose that can be used. We can also potentially treat intermittently with this chronic disease. But I also think there are going to be other therapeutics that are not as much in the limelight right now. There could still be anti-aggregation compounds that would be potentially safe. They may not be as powerfully efficacious, but they might decrease protein aggregation, in this example, A-beta. And I'd make one plug for the concept that with arguably 120 million people worldwide distributed around the globe with the Alzheimer process underway, we're going to have to think of some non-pharmacological treatments also. And all of us, there's not a doctor in the world that won't tell you that it's good to exercise, it's good to avoid smoking, avoid head trauma, et cetera. But we'll get much more sophisticated about things we can do starting at 30 that will make it less likely that we'll need a BACE inhibitor for 20 years or an anti-amyloid antibody.
So we focused so far on amyloid, and of course the other two pillars are tauopathy and tangles we've touched upon. We didn't talk yet about inflammation. Now, for years it was debated whether amyloid really triggers this disease. I think between the protective mutations in APP that block beta secretase cleavage, some early work you did, Dennis, showing that A-beta in classical cultures can induce phospho-tau. In our 3D neuroculture system that Duyan Kim did at MassGeneral, we saw that we could make plaques in a 3D system with human neurons, and if you wait 2 more weeks, endogenous tau turns into full-blown tangles, paired helical filaments. So I don't think there's much doubt that amyloid is one way to drive tangles, although you can get tangles with traumatic brain injury, with bomb blast, 5 bad seasons in the NFL. There are other ways to get tangles than amyloid, but amyloid is the insult that drives tangle formation over decades in Alzheimer's, and I think that's becoming more accepted. We also know that with amyloid and with cell death, there comes inflammation. Brad, I was hoping that maybe you could comment on some very insightful experiments going on in your lab with Teresa Gomez-Isla's work on the role of inflammation. And can we handle a brain full of plaque and tangles and not have disease if we can avoid inflammation? Much of the Alzheimer's genetics have pointed to inflammation, CD33, TREM2. I was hoping maybe you could comment on the 3 pathologies and how they relate to each other. And what do we need to hit first, and in whom?
Rudy, as you say, the brain is damaged in a lot of different ways in Alzheimer's disease. There are these plaques that accumulate throughout the cortex. There are tangles that involve a lot of the large neurons that project from one part of the brain to the other part of the brain. And the inflammatory cells, the microglia and the astrocytes throughout the entire cortical mantle are activated. And whether that's cause or effect, whether it's a damaged brain that leads to the activation, or whether that activation leads to a little synergistic cycle in which more damage occurs while those cells are trying to clean up the other types of damage, that's still a little bit uncertain. But the experiments of nature can help inform this a little bit. Teresa Gomez-Isla at MassGeneral has isolated some very, very rare individuals who at autopsy, their brains had many, many plaques and many, many tangles, certainly enough that the neuropathologist would have looked at them and said, gee, this person must have had the symptoms of Alzheimer's disease. With this much damage, they must have been impaired cognitively. And yet, when we went back through the medical records, the people seemed to be doing really quite well. And when Teresa asked what it was that was different between the brains of patients who had this amount of damage but were clinically doing well versus the average Alzheimer patient who and I want to emphasize, these were very rare cases. Mm-hmm. The difference was the amount of inflammation that was present in the brains. And so it seems as if you have to accumulate a certain amount of damage in order to develop the cognitive symptoms. That damage might be inflammation, it might be tangles, it might be plaques, it might be vascular disease, which is, as Dennis pointed out, one of the reasons why brain health, why exercise and diet are really a critical part of avoiding the clinical syndrome of dementia. And another piece of that, of course, is whatever damage is occurring as a consequence of the activation of the astrocytes and the microglia. So I think you're right. I think as time goes on, that's going to be yet another target. And as I think about the disease process in terms of these sort of 3 phases, this preclinical phase in which amyloid is accumulating and tangles are beginning to develop, and then the tangles spread out across the cortex, affecting more and more of the brain, causing more and more symptoms that are sufficient to bring the patient to the doctor. And then finally, at the point where a patient classically would be diagnosed with dementia, fills up the brain and has this big inflammatory result in the neuronal loss, it may be that the treatments for that last phase are largely against the inflammatory cells, and the treatments against the earlier phase will be directed against amyloid, and just as we've been talking. So I think that there's this incredible future of Alzheimerologists in the clinic, being able to have a whole array of different therapeutics to begin to treat patients at these different stages.
So Brad, you're just giving us kind of a view, just as we get into the last 10 or 11 minutes, you've given us a view of the future of treating a patient early for amyloid, or really a subject early before symptoms for amyloid, should you see higher levels in the brain by imaging. If there's tau pathology, perhaps using immunotherapy. There are multiple companies now developing immunotherapies to stop tau spreading. Kind of thinking like amyloid's like the match that lights the fire of tangles and inflammation fanning the flames. I just wanted to move to Roger. Roger, do you agree with this vision that we're hitting amyloid and tangles early, but in a patient with full-blown Alzheimer's, you really need to address almost an innate immune response, neuroinflammation by friendly fire, killing your own neurons? What do you think?
That's a very interesting question. I think some early neuropathological studies in patients that had received the active vaccine, very interesting in 3 or 4 cases, also few cases, but very interesting. The amyloid was basically gone in these cases, but they didn't stop on the in progressing with their dementia symptoms. So what that means is that to me, what that means is that the biology of antibody-mediated amyloid removal is working both in early brains and late brains. It's just the biology that is working, but the brain in the late stages is not plastic enough, is not healthy enough to basically regain functions. So I think it's very important to start with the anti-amyloid and maybe also the anti-tau treatments early on in order to hit the brain at a stage at which it still can regain functions again. And maybe what we're seeing there is actually training of new functions that had been lost before. Could be possible that there's 2 phases of this treatment. First is to reduce the amyloid and the associated toxicity, and the second phase, after the toxicity was removed, is training of functions, rehabilitation types of treatments. And so with respect to the inflammation, of course, there had been some very disappointing clinical trials with anti-inflammatory treatments, COX inhibitors and other things. On the other hand, there's also some encouraging results in large epidemiological studies where patients are looked at who take chronically anti-inflammatory drugs for other diseases. It seems to be that these people have a lower incidence of getting Alzheimer's disease. So even though I do agree with Brad's approach here, there's also a possibility that there could be protective effects of early anti-inflammatory treatments.
Yeah, please.
Just an add-on comment. I think if you look at any mature area of treatment for any disease, cardiovascular disease, for example, you don't give statins to somebody who's in heart failure, right? I mean, you can. You treat them very differently in heart failure than you do in the early stage. Right. I think in multiple sclerosis, we're starting to see a shift we focused on relapse and remitting, and now we're starting to see a shift to secondary progressive. So every mature disease, the way you treat the early stage is almost always different than the later stage. So I think that's the rule, and that's what we're going to see in Alzheimer's.
Like,
just one comment about inflammation. I think we have to be a little bit cautious, and the role is somewhat more, it's clearly quite complex. If you look at TREM2, which is a gene where a loss of function mutation, essentially, TREM2 is involved in regulating microglial phagocytosis. It's a loss of function mutation that increases the risk of Alzheimer's disease. So there we've got an example of what is sort of sometimes referred to as an inflammatory response, but it appears based on the human genetics to be actually beneficial, a required part of the process. So I do think the role of inflammation, or at least the immune system in Alzheimer's disease, is complex. There's elements of it which are clearly protective based on the human genetics. But as Brad's work has shown, there may be other elements that become damaging as the disease progresses, particularly potentially enhancing the spread of tau pathology.
we know on microglial cells, if TREM2 is keeping them more phagocytic, clearing amyloid, CD33 is the yin and yang of that. CD33 is turning microglial cells more into cytotoxic cells, free radicals, cytokine production. But in terms of targeting inflammation, I think you raise a great point that we don't want to turn off innate immunity in the body as we target innate immunity in the brain. That's our first line of defense. You won't live very long. So it's going to be quite challenging. And it makes it look like these early trials with ibuprofen look pretty naive, given how sophisticated we're going to be hitting microglia and sparing the rest of our innate immune system. Yeah, Dennis, please.
I think it makes the point that the best therapeutic is going to be for the earliest feature of the disease. So while I very much believe the innate immune system plays a major role in propagation of the disease and what my colleagues have said, it would be nicer to get upstream of that. And we like to believe at this moment in time, given the clinical validation we're starting to see, that A-beta in the Alzheimer's case is that. And what this really tells us is that we can ask ourselves why it took us so long to get to this, what we all agree is an exciting therapeutic moment. It took us 30 years or more with lots of battles and interesting things. If you look at the Parkinson field now, I think it's likely that the genetic data and the histopathological data come together the way they did for amyloid and Alzheimer's and suggest that synuclein regulation or dyshomeostasis is something that the pharmaceutical industry needs to target very thoughtfully and carefully. And we know they're doing it. And your company is working hard on synuclein immunotherapy, and others are here too. So I would just make the point that we don't need to take as long as we did to get to a real glimmer of hope in the Alzheimer field. We can see what the biology and the pathobiology are telling us. And synuclein, I think, would be a fantastic target. Whenever you have both a wild-type protein that builds up in neurons and causes disease, idiopathic PD, and you have mutations in that cause an aggressive, accelerated form, then you've got what we've come to discover in the Alzheimer field.
I think the other good news, I mean, there are companies out there like Abide, AZ Therapeutics, who are taking other approaches to hitting inflammation. We've seen cannabinoids can help. So I mean, there are going to be some out-of-the-box thinking there in terms of chilling out our microglial cells in the face of Alzheimer's pathology as we age. So we have just a few minutes left. I'd like to go down the line here and take about 30 seconds to a minute for your final closing statement. Yes, final closing statement, a vision for the future of treating and preventing Alzheimer's. Start with Brad.
Well, it's an incredibly exciting time. And I think that after really decades of struggle, we're just at the point where we're going to be able to offer our patients and those individuals who have Alzheimer's disease but not yet manifested the clinical symptoms of them some real hope and some real opportunity to stop that disease process and keep it from progressing.
Roger?
Very exciting times. Significant changes in the way how we're going to diagnose disease within the next 5 years. We're moving from clinical diagnosis to molecular diagnosis, molecular classification of disease. And so we're going to have a toolbox for specific agents that identify disease, and that will trigger the particular treatment decision, also with very highly selective compounds. And I think within the next 3 to 5 years, we're going to see the first disease-modifying drugs targeting amyloid on the market.
3 to 5 on the market?
Or at least for approval for marketing.
Okay. Mike?
So I share the optimism I said already. I do think we're very close now to having disease-modifying therapies that are available for treatment. I would just echo the point about the need for early diagnosis. We really do need a quantum shift in the way we do diagnosis right now. The majority of patients in the US and in Europe are diagnosed relatively late in the course, so that clearly has to change. It's partly about the kind of molecular toolbox that Roger and others have mentioned, the amyloid-based diagnostics, tau-based diagnostics, but I also think there needs to be education of patients and doctors. In particular, we need to really work to remove the stigma of Alzheimer's disease, which is, I think, one of the major reasons why patients don't seek help early in the course. And I think that's the other side of managing the Alzheimer's disease as a problem, is trying to get this quantum shift in the way we deal with patients, and particularly with diagnosis.
Yes, thanks. And Dale?
Yeah, I would say it's a three-legged stool. We've three things have to come together, and I think two of them are there. We have one left. As my colleague said, remain focused on early Alzheimer's disease. We now have therapies. We cannot forget that the diagnostic, the imaging, and biomarker tools have got to keep pace, or we won't win this battle. And finally, public awareness and public funding and funding biotech and pharma, I think, will be reinvigorated. There's the need, as you point out so rightly, for other new targets. We have to remain focused on all three of those to ultimately succeed.
That last point is crucial. Public funding, beautifully illustrated by Newt Gingrich's op-ed piece in the New York Times just a few days ago. This is an absolutely bipartisan, apolitical story. We got here, I think, a little too slowly after 30 years because of federal taxpayers' largesse. And we need to get back to that. And if you ask anyone what's the most important thing to them in life, it's keeping themselves, their children, their parents healthy. We need to put much more money into public funding, and that's going to help private funders do a lot more for us.
Well, I love the enthusiasm, I love the optimism, and I want to thank you all for a very informative and spirited discussion. So thank you.
Thank you for a great job.